Hi everybody. In this unit we are discussing carbohydrates. So for those of you following along in Voigt, and Pratt, we are primarily focusing on sections 1 and 2 of chapter 8, which covers monosaccharides and polysaccharides. Section 3 of this text covers glycosylated proteins, which we're not going to go into detail for this course specifically. But if you are interested in learning more about the role of glycosylated proteins, think about taking advanced biochemistry with me next year. So in class, we'll then apply the information from this lecture to complete section 13 from our workbooks. Okay. This section will allow us to practice recognizing and describing the structural and functional diversity of carbohydrates, as well as allow us to describe how carbohydrates interact with proteins and explain the consequences for these interactions. As always, these in-class activities enable us to further develop our ability to identify and utilize the diverse backgrounds of our teammates in order to solve interdisciplinary problems. So as we're working through these activities in class, don't stress out if you don't have the answer for every question. A pivotal component of the learning process is recognizing when we can rely on our colleagues' expertise to help us navigate a novel and unique problem. All right, so let's go ahead and get started. Here are the learning objectives for this unit. Again, guys, feel free to pause here and print this slide so you can check off each objective as you follow along with the video or as you read through the chapter. After watching the video and after we go through the in-class activity, do come back to these learning objectives and make sure that you understand what it is that I'm asking of you. If you don't, come see me during my office hours or make an appointment and I'm happy to meet with you. Carbohydrates are often referred to as saccharides. These are, just like we see with protein and nucleic acids, are also biological polymers. The single carbohydrate unit is referred to as a monosaccharide, where two monosaccharides covalently linked together through a glycosidic bond is called a disaccharide, such as we see in sucrose. And where we have a chain of 3 or more monosaccharides, we call that a polysaccharide, as seen with starch. Carbohydrates mainly function as the energy source for the cell. However, they can also serve as structural materials and in cellular recognition or communication. Monosaccharides are themselves aldoses or ketoses, depending on the location of the carbonyl carbon. If the carbonyl is located as the terminal carbon, it is termed an aldose. If it is located at the second carbon, then we call it a ketose. Now, based on the number of carbons and the stereochemistry about the various chiral centers of both aldoses and ketoses, we can have a wide array of monosaccharides. As we can see in this figure here, all of the aldoses seen in nature are derived from D-glycerolaldehyde, where the D designation comes from the stereochemistry about the second carbon of glycerolaldehyde. In nature, we see that the L form of these sugars are very low in abundance, and therefore our enzymes are not evolved to breaking down the L sugars. Bacteria, however, have taken advantage of this phenomenon by utilizing the L isomers of sugars within their cell walls in order to increase their resistance to the host's natural immune system. Now, while I do not expect you to commit all of these sugars to memory, you should be able to recognize the aldoses ribose, glucose, mannose, and galactose, as these are commonly used in cellular metabolism, and we will surely see these sugars again. Ketoses, like aldoses, are derived from a common starter. In terms of ketoses, they are derived from dihydroxyacetone, and the D naming is derived from the carbon 3 of D-erythrulose. The important ketose to recognize here is D-fructose. An interesting feature of monosaccharides is their ability to spontaneously form cyclic isomers. With aldoses, one of the oxygens of the alcohol in the carbon chain can react with the aldehyde carbon to form a hemiacetyl carbon. Based on the number of carbons within the ring structure and the stereochemistry about the hemiacetyl carbon, we can get further variety in the structures of these monosaccharides. With aldoses, we typically see six-member rings or pyrines, where the oxygen from the alcohol is part of the ring structure and the oxygen from the carbonyl carbon is reduced to a hydroxyl. When ketoses cyclize, we see a similar reaction, but instead of a pyrene ring, we typically see a five-member or furan ring. Just as we see in the aldoses, we can have different stereochemistry about the hemiketyl carbon, where the hydroxyl group is above or below the alpha or beta isomer ring structure. Just as we see in the aldoses, we can have different stereochemistry about the hemiketyl carbon, where the hydroxyl group is above or below to give the alpha or beta isomer ring structure. As you previously saw, the most stable ring structures we can form from monosaccharides are the five-member furanose rings or the six-member pyranose ring structures. Although larger ring structures can form, they are not the most energetically stable, so they're much less common. And rings smaller than 5 members are too sterically crowded. Monosaccharides themselves can undergo a number of chemical modifications, such as oxidation of the aldehyde group to form a carboxylic. When this happens, however, the aldose can no longer form glycosidic bonds with other sugars because the anomeric carbon has been oxidized. We can also have oxidation of the primary alcohol groups within aldoses to yield uronic acid. Uronic acids are often coupled with molecules to make them more water-soluble for secretion in the urinary system. Reduced forms of uronic acids are commonly used as artificial sweeteners. Another modification we see often are deoxysugars, such as we see in deoxyribonucleic acids. Also, amino modifications on sugars allows for covalent attachment of sugars to proteins or lipids, such as we often see on the plasma membranes of cells. A glycosidic bond occurs when you join two or more monosaccharides to make a polysaccharide. Glycosidic bonds occur between an anomeric carbon and an alcohol. Based on the orientation about our new chiral carbon as a result of the glycosidic bond formation, we can end up with either an alpha orientation, where the oxygen from the alcohol is oriented below the plane of the ring with respect to the terminal alcohol of the sugar, or a beta orientation, where the oxygen is on the same side or above the ring. Some common disaccharides you may recognize are lactose, where we have a glycosidic bond between the anomeric carbon of galactose, which would be carbon 1 for this monosaccharide, and the hydroxyl group off of carbon 4 of glucose. Because the alcohol group bound from the top of the ring, we see that this bond is designated as a beta orientation, hence why it is termed a beta 1,4 glycosidic bond. In sucrose, we have an alpha 1,2 bond between glucose and fructose. With maltose, we get an alpha 1,4 glycosidic bond between two glucose molecules. Cellulose is formed by a beta 1,4 glycosidic bond between two glucose molecules as well. And finally, isomaltose is formed through an alpha 1,6 glycosidic bond between two glucose molecules. One important thing to note is that in some of these disaccharides, we still have a free anomeric carbon in which we could bind more monosaccharides, as indicated by the green box. Sugars containing free anomeric carbons are called reducing sugars because the free aldehyde group that is in equilibrium with the cyclic form of the sugar is able to reduce mild oxidizing agents, and therefore we could bind more monosaccharides to that anomeric end. However, if the anomeric carbon of both monosaccharides comprise the same glycosidic bond as we see with sucrose, then there are no more anomeric carbons, and we call these sugars, such as sucrose, non-reducing sugars. And we cannot add any more monosaccharides because there is no open anomeric end. When we have 3 or more monosaccharides bound together through glycosidic bonds, we now have a polysaccharide. A common polysaccharide is cellulose, which is comprised of glucose molecules through the beta 1,4 glycosidic bond. This polymer, which is found in the cell walls of plants, is very strong, rigid, compact, and is water insoluble. This is due to the beta 1,4 linkage.
Because of the beta-1,4 linkage, we end up with a staggered chain conformation in the polymer, which allows for a very tight network of hydrogen bonding and compacting of multiple chains to form sheets. An interesting side note, vertebrates themselves do not produce enzymes capable of breaking down these beta-1,4 linkages, and therefore rely on the enzymes from the gut bacteria to digest these polymers. Another polymer of sugar found in plants is starch. Starch for plants is their long-term energy storage of glucose. Starch is comprised of amylose and amylopectin. Alpha amylose is formed through the alpha-1,4 linkage between glucose molecules. But because of the alpha orientation between this linkage, amylose tends to create a coil structure versus the pleated sheet structure seen in cellulose, which is due to the beta linkage. Amylopectin is similar to amylose, but with additional periodic alpha-1,6 branches as well. Starch can be digested by animals with the enzymes amylase, which is found in the saliva, as well as with alpha-glucosidase, which is found in the gut. To break down the branch points of amylopectin, specific debranching enzymes are used, which we will revisit this when we talk about gluconeogenesis and glycolysis in more depth. Because of the branching points on amylopectin, we have multiple non-reducing ends that are free to bind new glucose molecules or can allow the cell to break off multiple glucose molecules rapidly if needed. Glycogen, which is the main polysaccharide for glucose storage in animals, is very similar to amylopectin in plants. But instead of branching points at every 24 to 30 glucose residues, we can have branch points at every 8 to 14 residues. So I ask you this, why do you think animals have evolved to increase the number of branches for glycogen in comparison to plants with amylopectin? So think about that, and we'll chat about it in class. Again, our in-class activity for this unit is going to allow us to practice recognizing and describing the structural and functional diversity of carbohydrates. In addition, we will explore how carbohydrates can interact with proteins and explain the consequences of these interactions. See you then.